Welcome to CognitionChunks. This information is for entertainment only. I am not trained in genetics. Is this useful? Is this nonsense? Join me as I attempt to learn from GPT. Here is Designing PhytoSynthetica. Episode 6, Basics of Genetic Engineering and Synthetic Biology. Genetic engineering and synthetic biology represent two interrelated disciplines at the forefront of modern biotechnology, driving innovations that span across healthcare, agriculture, environmental science, and industrial manufacturing. While both fields manipulate the genetic makeup of organisms to elicit desired traits or functionalities, synthetic biology extends this paradigm by aiming to redesign and construct new biological parts, devices, and systems that do not exist in nature. This section provides a foundational overview of the principles, techniques, and applications inherent to genetic engineering and synthetic biology, laying the groundwork for understanding their pivotal role in advancing science and technology. Genetic engineering, the art of DNA manipulation. Genetic engineering involves the direct manipulation of an organism's genes using biotechnology. It encompasses a range of techniques used to modify the genetic structure of cells, including the addition, removal, or alteration of specific genes or genetic elements. The primary goal is to endow organisms with new properties or enhance existing ones, such as increased resistance to pests in crops or the production of therapeutic proteins in bacteria. Key techniques. Recombinant DNA technology. This is the process of cutting and recombining DNA fragments from different sources, often involving the use of restriction enzymes to cut DNA at specific sequences and DNA ligase to join DNA fragments together. This allows for the introduction of new genetic material into an organism's genome. CRISPR-Cas9, a revolutionary genome editing tool that allows for precise, targeted changes to the DNA of living organisms. CRISPR-Cas9 uses a guide RNA to direct the Cas9 enzyme to a specific sequence in the DNA, where it makes a cut, allowing for the addition, deletion, or replacement of DNA sequences. Transformation and transfection, methods used to introduce foreign DNA into a host cell. Transformation typically refers to the uptake of DNA by bacterial cells, while transfection is used for eukaryotic cells. These processes can be facilitated by various methods, including electroporation, chemical treatments, or viral vectors. Synthetic biology, engineering life. Synthetic biology builds upon the foundations of genetic engineering to not only modify existing organisms, but to create new biological systems and functions from scratch. It applies engineering principles such as standardization, modularity, and automation to biology, aiming to make the design and construction of biological systems more predictable and scalable. Core principles. Standardization of biological parts. Synthetic biology promotes the use of standardized biological parts, known as biobricks, which are DNA sequences with defined functions that can be assembled in different combinations to create new biological circuits and pathways. Design, build, test, learn cycle. A systematic approach to synthetic biology that involves designing a biological system, building it by assembling genetic parts, testing its functionality in a living organism, and learning from the outcomes to inform subsequent designs. Synthetic gene circuits, the construction of gene networks that function analogously to electronic circuits, enabling the programming of complex behaviors in living cells, such as logic gates, oscillators, and switches. Applications. Both genetic engineering and synthetic biology have wide-ranging applications that address critical challenges in various fields. Healthcare, production of therapeutic proteins, development of novel gene therapies, creation of synthetic bacteria for microbiome engineering, and design of rapid diagnostic tools. Agriculture, development of genetically modified crops with enhanced yield, nutritional value, and resistance to pests and environmental stresses. Environmental sustainability, engineering microorganisms for bioremediation, bioenergy production, and carbon capture, development of bio-based materials as alternatives to petrochemical-derived products. Industrial biotechnology, use of engineered microbes for the sustainable production of chemicals, fuels, and materials, reducing reliance on fossil resources and minimizing environmental impact. Conclusion. The realms of genetic engineering and synthetic biology offer profound potential to revolutionize how we interact with and harness the capabilities of the living world. By rewriting the code of life, these disciplines open new avenues for innovation, addressing some of the most pressing challenges of our time. As we continue to explore and expand the boundaries of what is possible in biology, ethical considerations and responsible stewardship will be paramount to ensure that the benefits of these technologies are realized in a manner that is safe, equitable, and sustainable. Thank you for joining us for another Cognition Chunks. Please like and subscribe.